Hi everyone, my name is Felipe Vaz Peres. I am an undergraduate student in biotechnology at Federal University of São Carlos in Brazil. And in this presentation, I'm talking about your preliminary results of your analysis of the novo transcriptome assemblies in sugarcane. But before I start talking about your preliminary results, I will do a background of my main project. My main project is to do the inference and functional annotation of the sugarcane pan-transcriptome. And what is a pan-transcriptome? Briefly, we can describe the pan-transcriptome as the union of all the RNA present in a species. So here we can see that we have the transcriptome of the cultivar 1, transcriptome of the cultivar 2, and transcriptome of the cultivar 3. And these transcriptomes have exclusive accessory and core genes. So the union of all of these transcriptomes can be called as the pan-transcriptome. And to do the inference of a complete pan-transcriptome, we need to have a lot of transcriptome assemblies as possible. Searching literature, we found 17 studies of the sugarcane transcriptome. We have only 8 transcriptomes assembly available, and we have 9 transcriptomes assembly unavailable. In this case, we have the raw sequence datasets of the RNA. So with these raw RNA datasets, we can assemble the transcriptome for these studies. And after this background, we can see our preliminary results of your analysis of the novo transcriptome assemblies in sugarcane. We create a pipeline to assemble transcriptomes from the raw RNA datasets. In the first step of our pipeline, we need to download the raw reads of the studies. Then in the next step, we need to convert the SRE format of the reads into FASTQ format. And to do that, we are using SRA toolkit. Once the reads are converted into FASTQ, we need to access the sequence quality. And to access the sequence quality, we are using the FASTQ-C. And then we need to do the quality trimming. And to do this step, we are using the bioenergy software. In this specific case, we are removing the sequences with quality less than 20. And we also are removing the adapters of the sequence, such Illumina adapters. After the quality trimming step, we can go to the contamination assessment and removal step. Here we are using the software acheive to access evidence of contamination in the reads. And if the reads have contamination, we use the bioenergy to do the contamination trimming. And if the reads don't have contamination, we can go to the transcriptome assembly. And here in these two tables, we can see the results of your contamination assessment and removal steps. The first table is the contamination predicted in the first step by acheive in this step. And if the reads have contamination, we need to go to the contamination trimming with bioenergy. So that happens here. But after the reads goes to bioenergy to do the trimming contamination, we can see that only 6 reads was removed for money, and none of the reads was removed for P. nicotinovanons. And this happens because we have a high rate of false positives in the detection of contaminants, or we are having low performance in the contaminant removal steps. And we are exploring alternative ways to detect and remove possible contaminants in this step, because it's a flexible step and it's possible to add other assigners to identify and remove contamination in the contamination assessment and removal steps. Once the reads are removed, we can go to the transcriptome assemblies. And in this step, we are using 2 sizes of chimeras to assemble the transcriptome with the Trinity software. As you can see here, both assemblies have unique genes, and in the assembly with chimera size of 25, we have 14 unique genes. In the assembly with chimera 31, we have 15 unique genes. But most genes are in common. You can see that in this intersection. And after the assembly of the 2 sizes of chimeras with Trinity software, we go to the step to create a non-redundant transcriptome with the software CD-HitYist. Here we created 2 transcriptomes with 2 different sizes of chimera, and then we created a non-redundant transcriptome And this non-redundant transcriptome is the merging of the transcriptome with camera size of 25 and the transcriptome with camera size of 31. And we did that to have a complete transcriptome as possible. After the step to create a non-redundant transcriptome with the CD-HIT yeast, we can go to the step to evaluate your assembly. To evaluate your assembly, we are using two softwares. We are using the Busco to check the completeness of your transcriptome assembly, and we are also using transrate to check some metrics of the assembly. And here you can see the results generated by Busco. As you can see here, the completed, the fragmented, and the missing Busco genes found in both assemblies are similar, and the same happens in the translate results. We have the original assembly in the first line and our translate metrics in the second line. To generate these comparative metrics, we used the sorghum bicolor as the reference transcriptomes, and we can see that both metrics are similar, and these two last metrics are really important because the CRBB metrics represents the CRBB reads, that are the number of reciprocal best reads against the reference, and a high score indicates that a large number of real transcripts have been assembled. And to do a summary of this presentation, our pipeline semi-automatically generates the novo transcriptome assemblies from raw RNASeq datasets of public repositories. The completeness and quality metrics generated by Busco and translating your assembly are similar to the original assembly. And our pipeline also includes a step to look for possible genetic material from contaminant organisms in the reads and remove them. And we are working to improve these steps. So if you have any suggestions or questions, please send me an email. Thank you for watching.